In the previous two lectures, we've talked about replication of DNA and transcription of DNA. Today we're going to talk about taking what is transcribed from the DNA by the messenger RNA and translating it into a protein. Gene expression leads to making proteins or protein synthesis. The genetic code consists of triplets. A triplet code is a set of 3 nucleotide units in a messenger RNA molecule, and that triplet is called a codon on messenger RNA. There are 65 different messenger RNA codons that exist. 61 code for particular amino acids. There's a redundant code sometimes, and some amino acids have numerous code words. It provides some protection against mutations. And there are stop codons that signals the end of a polypeptide being formed. So here we have messenger RNA codons. So for example, if we have UUU on messenger RNA, it codes for phenylalanine. The code UUC also codes for phenylalanine. If you come across a set of triplets on messenger RNA that say UGA, it's a stop codon. And AUG is a start codon. So there are 20 amino acids that make up all the proteins that exist, and of those there are 64 messenger RNA codons that code for those particular amino acids. Let's take a look at how that all wraps up together with making proteins. A transfer RNA is a little bit different character. It transports amino acids to the ribosome. It has a boot-like shape to it, and the amino acid binds to one end, and opposite that end has what we call an anti-codon. So the messenger RNA has a codon. This triplet is CGG. The anti-codon on the transfer RNA that is going to complement this side is going to be GCC. So the transfer RNA that matches up to this codon is going to carry the anti-codon GCC. And when we look for the messenger RNA CGG, it's right here, and it's one of the codons that codes for arginine, which if we go back over here, arginine is the amino acid that's going to form a protein. So the order of the messenger RNA codon determines the order that the transfer RNA brings the amino acid in as well. So let's look at some pictures of what this would look like. So we have a transfer RNA, and in that transfer RNA there's going to be an anti-codon. That's what this shows right here. And the amino acid, this is arginine up here at the top. That amino acid is going to get dropped off during translation and is going to become part of a polypeptide chain. So the DNA, just to recap, the DNA is in the nucleus and it's a double helix, and we are going to transcribe that. So when we transcribe this section of DNA right here, it transcribes to these particular codons of messenger RNA. The messenger RNA is then going to go out to the cytoplasm through a nuclear pore to the cytoplasm. It's going to find a ribosome. It's going to hook up with the ribosome and be translated. And it's going to be translated by the ribosome expressing the need for these particular anti-codon segments. So as it goes through and it reads CGG, the anti-codon GCC will be the one that pairs up with it. It pairs up with it and drops off its amino acid. The next triplet that goes through is UAC. UAC is going to need AUG. That transfer RNA is going to come along, drop off its amino acid, and those two amino acids will be bonded together, and the two anti-codons are going, the two transfer RNAs will go back out into the cytoplasm and pick up a new amino acid and be ready for use again. And this continues until the entire segment of messenger RNA has been translated into a protein. Ribosome and ribosomal RNA, free or attached to the endoplasmic reticulum is where we're going to find it. It's made up of 2 subunits, a big one and a small one. It has binding sites for the messenger RNA and 3 transfer RNAs. And the binding sites help the complementary base pairing between the transfer RNA anticodons and the messenger RNA codons. Brings amino acids in a line in a specific order for a polypeptide to form, because the order of the amino acids determines the makeup of the protein. Several ribosomes can move along the same strip of RNA at the same time, so multiple copies of a polypeptide can be made. The entire complex is called a polyribosome when it's all put together. So here we have the large subunit and the small subunit of the ribosome, and this is where the messenger RNA gets translated. Notice that can happen several different places so that we can have multiple copies, and it just moves along the messenger RNA and continues to add the amino acids as the transfer RNAs bring them. The transfer RNAs come off, go back out and find another amino acid, and they code for that specific amino acid. So if they drop off arginine, then they're going to go back out. The transfer RNAs will go back out to the cytoplasm and pick up another arginine. So translation requires 3 steps. The first step is called initiation. This requires energy. Initiation factors assemble components. They always start with the start codon, which is AUG. P, peptide site, A, amino acid site, and E, exit site. Elongation is what happens next. This requires energy as well. And then there's a termination step that happens during translation and making a protein. This is where the stop codon is reached, and that particular protein stops being formed, and it requires a release factor too, so that it's released from the messenger RNA. So the initiation stage, we have a small ribosomal subunit binding to the messenger RNA and an initiator transfer RNA with the anticodon UAC pairs up with the messenger RNA that holds the start codon AUG. The large ribosomal subunit completes the ribosome. The initiator transfer RNA occupies the P site, and the A site is ready for the next transfer RNA. So we have the E site, the P site where it starts, and the A site which is ready for the next one to come in. The transfer RNA transfers an amino acid as it approaches the ribosome and binds to the site A. And elongation occurs by this continually happening until the whole sequence of messenger RNA has been translated and a stop codon is reached. During the termination phase, we come in contact with a messenger RNA that has a stop codon in its sequence, and a release factor binds to the site. The release factor hydrolyzes the bond between the last transfer RNA and the P site and the polypeptide, releasing them, and the ribosomal subunit is going to dissociate at that point. And what you have now is a messenger RNA that has been translated, and you have a protein that has been formed because of that translation. So far we've covered DNA replication, transcription of messenger RNA, and translation of messenger RNA into a protein, a long polypeptide chain. On the test, you need to be able to tell me the steps of replication, transcription, and translation. Remember that DNA replication only occurs during the S phase of interphase and only happens when the cell is ready to divide. Translation and transcription happen frequently within a cell because that's how a cell makes the proteins. So in transcription, it's going to happen in the nucleus as the DNA is transcribed by a messenger RNA. That transcription piece, the messenger RNA, it then goes through a nuclear pore into the cytoplasm where it hooks up with ribosomal RNA subunits and is then translated into a polypeptide chain or a protein with help of the transfer RNAs that hold the anticodons that are complementary to the messenger RNA codons.